microbes that can create really interesting materials. We may get to the point where we can plant a seed and have a building.
Are we going to put GMO bacteria in the ground, Martyn?
The materials that you're working with have agency. Well, the kind of guerrilla living architecture. I'm a better academic writer than I am a science fiction writer.
Hello, everyone. Welcome to Solarpunk Manifesto. Today, we are at Northumbria University in Newcastle, and we're recording an episode with Martyn Dade-Robertson, who is a professor of emerging technology and an academic lead at the Hub for Biotechnology in the Built Environment, which is an interdisciplinary research hub between Newcastle University and Northumbria University, which does a lot of really cool stuff. Most of it interdisciplinary with applications of bio stuff in architecture. And it's pretty difficult to explain what exactly it encompasses. So one of the first questions that I have for you, Martyn, is when people ask you what you do, how do you respond?
So you're going to start with an easy one then. So what do I do? So my background is in architecture. So I used to design buildings, although I haven't done that very much recently. So I have a sort of joint degrees. I have degrees in architecture, computational technology as it relates to the built environment broadly, and then synthetic biology. So it goes across the scales of everything that you can possibly design. So I like designing things, but I like designing things now with biology, which means starting at the nano and working all the way up to the built environment. So my interest is in new materials, biological materials and biological fabrication specifically. So one of the mantras of the centre and the group that I run within the centre is, can we grow a building? So that's what we're attempting to do from scratch, using cells, but still mixing cell technology with other kind of technology. So that's what we're doing more broadly. And then the hub, which I founded now 5 years ago, looks across different themes connected to biotechnology in the built environment. So looking at construction, so living construction, the kind of thing that I've talked about, but also looking at the interaction between buildings and their natural environment, particularly the microbiome we focus on. And then also the idea that buildings could have their own metabolism, so are able to process wastes themselves. They actually make useful things out of things that they would usually get thrown out, right? So buildings no longer become an input for stuff and then an output for stuff. They're able to metabolize things, and so that's the third broad theme. And I had a role in kind of overviewing all of that stuff really and putting the strategy for the centre together.
So how come you became interested in it in the first place? Because as an architect, you just build stuff, but you decided that growing stuff is cooler, more interesting. What particularly attracted you to it? Yeah,
it was, I can pinpoint this precise time and moment that I decided this. So it was Newcastle University were involved in the iGEM competition, the international competition for genetically engineered machines. So people in synthetic biology know about this. It's one of the kind of foundations of the world of synthetic biology. And a group of students had entered the competition with a bacteria that they proposed could swim into cracks in concrete and then repair concrete buildings effectively, these very micro cracks that are very hard to treat. And it was published on the university news site because they'd done very well. I think they got a gold medal. And my thought was, this is really cool to start with. And then secondly, why are we not doing this? Why is no one from my department doing this? Mm-hmm. And then I kind of realized that there wasn't that many departments in the world at all doing this kind of thing. And that seemed to me to be cutting edge research. And I'd been doing lots of digital technology research, and some of that felt quite old hat, actually. Well, I wanted to, if I could have been in computing, I wanted to be in computing in the 1960s and 70s.
Ah, okay. So you like to be at form.
So yeah. So synthetic biology, it feels like it was about that moment, right? So it was about to do something. So I then persuaded my wife, who's a molecular microbiologist. That's very helpful to have, to let me into her lab to mess about for a summer, which we did.
Okay.
And then the moment, it was a real hobby project, but I was interested actually in whether we could get the bacteria to produce calcium carbonate, the basis of a cement. And there'd been some research in this already, so we were not the first to that by any means, but we did manage to get these little calcium carbonate crystals. And I remember the email that my wife Mung sent me with one of these little crystals. And so one of the things about microbially induced calcium carbonate crystals is they can quite often have quite distinctive forms. these images, I mean, electron microscope images are amazing anyway, but these images were just phenomenal, right? So for me, there was kind of microbial architecture, tiny, really small. So that became then something we had to get over, but that was the moment. So then from then on I was hooked, and then it became essentially my research. My entirety of my research life became about that.
So you evidently became interested in it purely because of how exciting the research was. But did you at any point start becoming more aware of the problems in the construction industry that you wanted to contribute to fixing, alleviating by means of your synthetic biology research, or was it just kind of something that was on the background?
No. So those things came together. So the microbial cements thing was the starting point for that. So concrete is the sort of number one problem in the built environment, really, in terms of carbon emissions and everything else. So sorting the concrete issue is still an unsolved problem, still an incredibly versatile material that we use extensively. There are biological ways of dealing with that. There are biological cements, and we work on some of those in our lab at the moment, but there's nothing of the scale of concrete. My first project was, I figured I couldn't take on the problem of concrete initially, and I ended up doing a master's in synthetic biology. So that was the first project that I did. We thought we'd start with building foundations. So my concept was that, so there's a real issue about removing earth in construction. You remove an awful lot of earth. Often you remove a similar volume of earth to the whole construction itself. Mm-hmm. And then you have to find somewhere to put it. And so that's a huge aspect of construction that we needed to get rid of. And so I had the concept that we could put microbes in the ground and get them to respond to mechanical changes in the environment, and then synthesize calcium carbonate, binding the soil grains together and making a self-constructing building foundation. So that was the theory there. The idea being that we could have this completely passive approach to the construction of a foundation. You don't need to dig a trench, fill it with steel and concrete. You could just run with what the ground can do. We called that project Thinking Soils, and it was quite an audacious one. Mm-hmm. It's 10 years of work on this now. We're just about getting some interesting data. But so that was really, we thought about that from the beginning, and about ways in which growth, particularly biological growth, is a more sustainable model for the way we fabricate materials than traditional forms of construction. So this has been going along the whole time. We're looking at ways in which we can have these very limited energy resource, in terms of energy and resource methods, to produce really complex materials. And that's what we're trying to get to.
And what about the materials that you're trying to synthesize in terms of its application scale-wise? Who are the research institutes that are investing in this kind of research? Are you experiencing any interest from the industry itself, the companies?
Yeah, industry is a tricky one.
Mm-hmm.
The construction industry is traditional and conservative for lots of reasons. We could come back to that. I think there is changes. I mean, so we set up the Hub for Biotechnology in the built environment five years ago now, and we did have a list of industrial interest, but I think it was pretty marginal. That's changing a little bit now, because there are lots of construction sector companies who have now quite tough sustainability goals, and they're looking at their bottom line, to be honest. So we do get approached a little bit more often. We get approached much more by startup companies. So there's a vibrant emerging tech sector of startups working in this material domain. Scale is a problem. And there are research centres internationally that do this kind of thing. TU Delft, for example, had a spin-out company where they're working with self-healing concrete, for example, which is the kind of stuff I was talking about earlier. And there are companies as well. So Maxim would be the main microbial cement company. There are a few like it. There's a startup. I hear that there's a startup in the UK in this area quite soon as well. So it's there. There's lots of interest. I think it's one of these things where you'll find most departments of built environment that have a strong sustainability agenda will have some kind of biomaterials part, but it's often only individuals. So when we set up the hub, it was recognizing that there was this research internationally, but there was no real centers for it. I think there are a handful of architecture departments that now have biological labs.
Yeah.
So in the UK, for example, there's the Bartlett's, the BioID program there has a biological lab. And we get approached by different groups to talk about how to set up labs, but it's still very unusual.
Yeah, and this is usually, are we talking about molecular biology labs, or is it usually really just for growing like mycelium or?
Yeah, the best microbiology labs are often enhanced kitchens.
Yeah.
There's like it's a sliding scale, right?
Yeah. Yeah, I think to our non-initiated listeners, the HBBE indeed offers a very unique combination of facilities in which you can both do the typical architectural tests for conducting any type of research in this field, but also has very extensive labs with everything you could possibly imagine for molecular biology work to perform the synthetic biology successfully, so that allows the HBBE to synthesize these two types of research into, and being essentially, I think there are only two places on Earth really that can do this in an academic setting, you and Boulder, Colorado.
Yeah, Boulder would be the other major group there. I mean, I guess that you could argue also MIT have got aspects of those facilities together, and they have most things. But so we organized, when we found the HBBE, we organized it in terms of facilities of scale. So we had a map that took us from the nano, so molecular biology, the Cat2 kind of lab facility, all the way up to my lab, which was the kind of cross between bench scale and microbiology, and then up to a building, the OHM, the experimental house. And so that provided us with a kind of end point for any materials and technology that we could then deploy within a kind of real, in that case, a kind of domestic or pseudo-domestic environment.
So basically, what you're saying is that HBB was set up like this by design, that you were really putting effort into the fact that you could do the whole pipeline, the whole funnel of researching things specifically for applications of biotechnology in the built environment in its broadest sense, as you usually point out, that it's not only that you have different types of biomaterials that we're talking about here. They either consist of living organisms, right, or facilitated by, et cetera, so there's a spectrum that we can explore. And I think this is a good point really to give our listeners a blitz of all the stuff that is happening here, because all these concepts, perhaps to Lola as well, sound a little bit like Greek. So we're going to go through the main points and just spend a few minutes talking about each of them, because you are one of the main contributors really in the world to a lot of these concepts, such as biodesign, biological architecture, living architecture. And you've also been at the forefront of really synthesizing a philosophy and a vision of how to work in these fields. So to start with, you became interested in the applications of biotechnology in the built environment. Would you call that living architecture? Would you call that biological architecture? What is the difference? What is your preferred term? It's like defining synthetic biology, isn't it? It's like there's no definition.
Yeah, no, and a lot of people are a bit territorial about the use of certain terms as well. It was interesting because the term living architecture has had a number of different iterations with different people. Yeah, absolutely. So I know, for example, there's Philip Beasley in Canada has the Living Architecture Systems group that we were part of. Actually, it was a network, but also very much his lab. And there's Rachel Armstrong as well, who has a project called Living Architecture, in a EU-funded project. So the term living architecture is a broad one. We did sort of form in that manner. Biological architecture is a bit broader. I mean, so I would say that for my interest, there's three steps to it, right? On the one hand, there is essentially research into the biological, sticking particularly with construction and materials, which is really my area.
Right.
On the one hand, we've got what I would say are quite relatively late stage kind of research on using biological materials, often quite traditional biological materials, so timber and different forms of limestone and other things, which have a biological origin to them. So and in a way, the whole history of architecture and the built environment is connected to that history, right? It's a history of biomaterials, and we can't ignore that because there's quite an important history there, and we still rely on a lot of those material sources. We are then interested in more advanced synthesis of biological materials that perhaps wouldn't exist in nature if we didn't synthesize them, mainly around microbes, so biological cements, also all of the kind of biological polymers that we can make, cellulose and so on, that you can either use as a material or more likely as a binder for other sorts of materials and wastes particularly. That would be our ideal. So we would use.
You mean for timber and for other biomaterials?
We can use those to treat timber. So we have projects where we're looking at new timber glues and timber treatments, but we can also, for example, then make use of agricultural byproducts. So my wife Mung works on a project with a company making composites with the waste from lime quarries and also hemp and other kind of agricultural waste into fiber boards. But we're using microbial processes to create sustainable binders for those materials, and we give them enhanced properties like fire retardants and other things. Then you get into the next stage, which is getting a little bit more futuristic, which is where we think about whether we can self-assemble large-scale structures biologically, so we can use the intelligence within cells to build things. And that would be, for me, so getting to an ideal. That's not necessarily, I mean, I guess the poster child for this would be a tree, right? You can put a seed in the ground and then you can construct something of extraordinary and exquisite complexity that's entirely self-assembled in relation to its environment. We have some middle ground projects where we say, for example, we have a microbial cellulose where the microbes will produce the cellulose, but we guide their growth through external means, in this case, kind of a robotic fabrication process. So we make use of that growth process, that process of self-assembly, but we push that material towards more complex things. And then the final step of this, the really future vision, would be not only the kind of self-assembly that you get from a tree, where we really could just program some cells and we could get them to self-assemble into structure and to distribute material in that way, but also they retain living or lifelike qualities, what are known as engineered living materials. And so they'd be capable of sensing, self-healing, responding, adapting to their environment, maybe even resynthesizing themselves after destruction or whatever. That's the kind of high end of things, but we don't then leave that. That's not the end. So we're not just working on the aggregates. We're trying to do the ELMs bit as well. Yeah. So we're doing all of those different things. And that's what I would call living construction. So that's the kind of gamut of what we're trying to get to.
And what is the reception by your fellow architects when you tell them about this kind of stuff? What do you usually get the side eye, like, oh, this guy's crazy?
It's a combination of, I mean, there is a group who are really into this stuff.
Yeah.
So usually I'm in the miscellaneous bit of the architecture conferences, but with at least 3 or 4 people who are deeply devoted to the cause. So it's a good experience from that perspective. From others, it's a bit of bemusement. I mean, I think there is a misnomer in a lot of architectural technology research, which is that we should wait for the technology to catch up to being constructible.
Yeah.
So if you've ever come across the technology readiness levels, you've got from level 1, which is the sort of stuff that pure science will do, and then it goes all the way up to sort of level 9, which is mission accomplished.
Yeah.
And there is a step at about level 7 where you start to do environmental trials or real trials of whatever technology that you're using. And I think there's a tradition, particularly in architectural research, of waiting for things to get to level 7, and they're deployable in the built environment. So when I talk and I'm showing images of stuff in a microscope, people are saying, well, yeah, but that's like material science. That's not architecture.
Yeah.
And so that comes up a lot. And that argument, and my counter argument to that, is that we can't wait for those materials to mature to this stage. And nobody's going to drive those materials forward if we don't.
Yeah.
They will be driven for aeronautical engineering or whatever. Those are the people who have a hunger for a palpable hunger for advanced materials, but we should also have a palpable hunger for advanced materials. And as we start pushing that agenda, we're not going to get that research anywhere. We're going to wait another, 100 years, and then we'll be in a position to start utilizing the things that we've acquired from elsewhere.
Yeah, we also talk about this quite a lot with Lola, that it's problematic that indeed the aeronautical industry is the one that is usually really interested in all of this bio stuff, just because they're sitting there at NASA and they're throwing money at anything that just has advanced, novel, in like living and self-deployable. Really, self-deployable is the word probably. So if you can just send off some spores to Mars and it will grow there, it's much easier. It saves a lot of money to some people. So they're throwing money at it. But here on planet Earth, the architects indeed sitting at the conferences, they're listening to you and they're thinking, well, iterative cuts on the carbon emissions and stuff like that, it's working, right? But no one is really because we have settled for a particular type of architecture. We are not trying to imagine new ways, which is really detrimental to the vernacular architectures and the use of local materials, right? And this is also what you speak of with using the waste streams from local farms and such, because there's really so much potential to bring it back and to own the materials and not just say, there are steel production facilities like in Brazil that we get our steel from. So we're going to build from steel here as well. And the buildings are going to look the same in Japan, in the UK, in Chile, and it doesn't matter. Sucks, but I hope we'll get there. I guess my next term in the Blitz is about biodesign. Where does biodesign feed into this? And is it really just a communication tool, or is it more than that?
No, I mean, I came to biodesign after discovering sort of biotechnology and synthetic biology and other things. And I think it's as much as anything about trying to find a community. Biodesign is an interesting one to try and define. I've just had a paper rejected because I didn't define biodesign well enough in the introduction.
Oh.
Which was like an impossible task, really. And so I have a biodesign book series, and we do at the beginning of that have a plate where I try and define biodesign, and essentially it's undefinable because so many people have different bits of it. Because it started off really being, if you go to a biodesign academic journal, it's mainly around the design of prosthesis and sort of devices that will be in the body and so on.
Yeah.
And then it was derived, I think Myers, the Myers book, the biodesign book is the one that most people would go to define biodesign. And it's essentially designing with biology as being distinct from, for example, biomimicry, copying or being influenced by biology, or there's an overlap there we might get to. But so designing with biology, it has on the one hand a sort of a technical lineage with synthetic biology, I think, but is about trying to think much more broad terms about applications which also have deep ecological understanding, social contextual understanding, which is what design gives you. So great designers are multifaceted people. They're able to understand breadth as well as depth of a particular subject and to create for it. And it's been, I think those who do biodesign kind of know that they are doing that. And it's helped a lot by, for example, the Biodesign Challenge, which we got involved in. I'm trying to set up an academic community in this area because I think there's some really interesting research coming out of different design schools that doesn't find its way into publications because it's difficult to do that, and because there are people who perhaps are not writing or thinking in the kind of way that's necessary for formal.
You didn't define biodidesign.
But I haven't defined, no, and I probably no accident that I haven't actually. Yeah, I don't know. I mean, I don't think there's a dictionary definition of it yet.
I agree. I think it shouldn't be so constrained by the definitions, and perhaps setting up you already have the paper call, right, for the Biodesign Challenge.
Yeah, so we worked with them. So we set up, I did three things. One was we worked with the Biodesign Challenge quite early on because that felt to me like the right community to work with, and setting up an extended network. So originally we created a kind of network in Europe as well that were related to the Biodesign Challenge. Set up a journal with Cambridge University Press called Biotechnology Design, which was there to try and sort of set really the parameters for academic scholarship in this area. So valuing creativity, originality, and new ideas, but also recognizing the need for rigor as well. So the journal was set up in a particular way to allow different audiences to submit to it. And then the book series for much longer form work, particularly from lots of PhD students who were working in this area. And that was the idea that there were some great PhD thesis. It was just sitting in university, deep in university kind of websites that deserve a little bit more exposure. And then putting it together to try to create a community of people who at least all agree that they're biodesigners. Even if we haven't tried to define it in any of those terms, but there is a community there. Definitely, we recognize each other in the room.
Yeah.
And so that's good enough at this point, I think.
Okay. In terms of biodesign and designing with biology, I think we have some questions about this, and that we might want to define, if not define, then just talk about, right? So when you design with biology, you can both still have it alive or not alive, or is it necessary that it's alive at the point of use? when you're designing it. And so you speak of, you use this concept of agential materials, so materials that have agency. What does that mean for biodesign and biological architecture?
Yeah, the first part of that is, it's another tricky one. When we think about materials, we just define the difference in our lab between engineered living fabrication and engineered living materials. So engineered living fabrication is where we utilize living cells, and we almost always are using living cells. There's an argument over whether cell-free systems can fit within this, and I think that is within the biodesign remit, but we tend not to, and mostly it's microbes. But the ultimate output of an engineered living fabrication is often a material that is then, if not dead, denatured, dormant, whichever way we want to go with this. We don't like killing things, but they are not alive anymore. And I think that's where we can see the most early impacts of those sorts of materials, because they are, once they are there and they're dormant, then you don't have to worry about keeping them alive, and you don't have to worry about infections or contaminations and all those things. And then there's engineered living materials, which might be fabricated through engineered living fabrication, but then retain their living properties afterwards. And we also make a distinction increasingly now between high and low metabolic materials. So materials that might have a lot of dormancy in them, but have that potential to come back to life again. We have a couple of projects that do that. And then the other question was about agential materials. So the idea of agential materials actually comes from developmental biology, and particularly an academic at Tufts University called Michael Levin. I would definitely recommend you get him on the podcast if you can. He's done it was a podcast episode that I heard him on originally. So he's a developmental biologist, and he essentially has three categories of materials. So there's inert materials, the materials that we're very used to designing with in our day-to-day lives, and as an architect I was brought up with, which are, there's no material that's completely inert, right? But materials that are stable and predictable for the lifetime of their use, right? So the table, for example, is made of that kind of material. Then there's been an interest in active materials, which are, I think the formal definitions of materials which are in a non-equilibrium state. Generally speaking, if you give them energy, they will do something, they'll change. So for example, shape memory alloys, where if you heat them up or you pass an electric current through, which heats them up, they can change their shape and form. Hygromorphic materials, which will absorb water and change their shape and form. And there's an increasing interest in these kind of active materials because you can use them in all kinds of different contexts. So you can engineer them to be programmatically responsive. Yeah. And there's a lot of work in that area, particularly in chemistry and the development of new materials. And then there's this agential idea, which is what Michael has introduced, which is that when you're working with living cells, they are not simply responsive, right? So it's not easy, even with a simple microbe, just to find its input and output. They will change. They'll often change their environment. They'll change themselves. They may change their morphology. They may grow or distribute in different ways. But they're also making decisions about their environment in some way. They're deciding what is the most favorable for them in that particular condition. And in his world of mammalian biology, these multiple cells will be programmed depending on their conditions. They'll make decisions whether to heal something, or in early development, whether one is going to be an eye or a brain cell and so on. There is this real complex interaction, and it's not always clear what the right sort of interaction with those things is. If you want those cells to do a certain thing, you want them to produce a certain material or take a certain form, the question then becomes, how do you persuade them to do that? Because they have their own agency. It's not something like with an active material where you can define an input and output. The programmability of an agential material is completely different. And that's what's kind of exciting. So even working in a biological lab, as you will know, things often go wrong. In fact, mostly they go wrong. And part of the reason they go wrong is because the materials that you're working with have agency. Yeah. And you have to kind of understand that agency if you're going to work well with them. And then there's a kind of ethical layer on top of that, I think, which is to say that if something has agency, then maybe you need to have a conversation. Now, can you have a conversation with a bacteria cell? I don't know. But you certainly have to recognize that the material that you're dealing with is not like a normal material, not the kind of the materials that designers are usually used to dealing with. This idea crops up a lot in biodesign, I think. It's called different things. It's reflected upon in different ways. But for me, it's a central idea in biodesign. It has to be a central idea.
Do you think we also need a different approach to educating even the undergrads or perhaps even children about the materials that, okay, this table made out of plastic or whatever fibers, it's not supposed to be so static. Do you believe that this is possible, that we achieve a world in which things around us are living, so we kind of live in a engineered forest that is accommodated to our needs, but it is still alive, and it's, you know, self-healing, self-cleaning, self-whatever, and it will maybe decide to change the color, and you respect that instead of being like, oh, my walls are rotting. You know how it is now.
Yeah, I think so. I mean, so there's two bits to that, right? One is just raising the imagination bar a little bit, just not being set in a particular form of things just because that's the way they are. And I would like to see a little bit more of that, that kind of creativity of thought brought into the classroom. It's interesting because I've been involved in my son's high school quite a bit, and it's really driven by the art teacher. Frankly, the kind of biodesign interactions that I've had have been driven by the science teachers as well, but the art teacher has been very keen, has come to a lot of our sessions, has brought students over to see our labs and so on. And so there's that level of thinking which comes from that world. I think there's also something quite fundamental that we need to be teaching, and I don't know what the right moment is to be doing this, whether it's at high school or university or whatnot, but teaching complexity. I think particularly in the way that we teach engineering subjects, it's often about simplifying a problem rather than complexifying it.
Yeah.
And so the key with agential materials is that they are really complex problems for which there is no single solution. We deal all the time with complex problems in the broader design world, right? So there's this idea of a wicked problem, which is a problem that has no optimal solution, but for which you're making payoffs and decisions that about what causes least problems or what creates the best out of the ingredients or the circumstances that you have. And I don't think we teach that complexity enough. And as an engineering task, it's not about simplifying that task into a single leverage point where you can design the optimal thing. It's approaching an entire ecosystem of a challenge domain and then working out where you leverage within that. We understand complexity a lot. We have lots of ways of modeling and understanding it, but I just don't think it's taught. Certainly not taught at high school level. I'm not sure it really is taught enough at undergraduate either.
Yeah, I feel like complexity problems is just overwhelming to a lot of people, and we don't like to struggle with it. And these days, we don't even like to be aware of it, because indeed the inherent complexity of things like climate change or things like agential complex materials is just too much to comprehend. But this is definitely something that perhaps we can address, and you, I think, are also very much dedicated to raising awareness through biodesign education as an umbrella term. As we've identified today, biodesign is not probably what you think. If you tell to a person, any person in the room, they say, oh, biodesign. It's just broadly about design, and there's somewhere biology in there swimming around. So I think we have initiated our listeners sufficiently on these terms. Yeah, it's not, but this is the complexity you have. So, you know, so I think now I'd like to have a crash course on what's happening here at the HBBE. And there's many projects, and one of them you already mentioned with Thinking Soils. And the way I would like to introduce these different projects to our listeners is by not just talking about the formalities about the genetic circuitry that you're engineering, but really thinking about the applications. And I think you very beautifully started explaining Thinking Soils by saying, well, you know what, we dig up all this ground to put concrete instead of it. But what if our ground was the concrete? What if we could just put the bacteria in there and make them basically solidify the ground, and it would function as the foundation for our building? Let's maybe start by talking about this, since we already touched upon it. Could it hold a skyscraper, this kind of structure? And what should our listeners be imagining? Is it that you literally put these bacteria in the ground, they solidify the ground, and you have something like concrete? Or is it going to be a smaller building? What would that look like in your vision?
So, no. We couldn't get a skyscraper. No. I can say that with I mean, for partly because one of the things we've been doing is, so we work with soil engineers, for example, and we've been doing modeling to look at if you actually map on the amount of bacteria that could reasonably survive in the soil, really all of the activity is in that top layer, right? So you don't get a lot of activity deep into a soil for lots of reasons. Our bacteria wouldn't survive in those contexts. I have to say that the Thinking Soils is an example for me of a project that through a big concept drives some quite early technology readiness at work. So what we have in our lab and we're about to publish on now is a mechanical sensing bacteria. So we do have a gene circuit that we can detect mechanical changes. You can imagine that in an application like, I don't know, something in soft robotics, for example. That additional sensing capability in a biological system could be useful in maybe a fabrication context. So the way we demonstrated, we have a jelly essentially that we grow a three-dimensional culture of bacteria in, and then we can load the jelly and you'll see parts of the jelly go white with calcium carbonate. So you can imagine a kind of self-responding structural something or other. And I suspect we don't really know what the application is going to be, but it's driven some really interesting research. And we've had lots of people come to us with those ideas. Microbes are used in soil remediation and strengthening though. So one of the areas that we've been investigating is preventing liquefaction. So if you've ever come across when earthquakes happen in certain soil conditions, because of the vibrations, the soil particles effectively slightly separate, and the soil particles will go from being very solid to being liquid, and you get landslips, and this can be a real problem. Even a relatively small amount of calcium carbonate deposition in those contexts can mean that you get a much stronger soil in those conditions. So actually another spin-off from this we've got is looking at soil improvement more generally. You don't need the intelligence of a biological sensor, you just need to find the right bacteria to put into the soil to do that job for you. So I think that's more likely to be a kind of an early stage application of that. And as I say, other groups are working on soil improvement generally using these microbes. We want to try and, I guess, to really understand completely the biological process that's going on and have ways in which we can turn it off and on. So that's been the other side of this project. And that might be useful in, I don't know, building embankments, for example, where we could treat the soils, we could do certain things to them without significantly having to move or add in additional materials into those contexts.
Or fortifying soils against erosion and stuff like that.
Yeah, absolutely.
So when we're talking about this, it begs a question probably for people who aren't synbio heads, you know, are we going to put GMO bacteria in the ground? Martyn, is that going to be happening?
Is that going to happen? I think we will. I mean, I don't know whether we will. We're not allowed to.
I hope as soon as possible.
But I think we will. I mean, there are different questions over ethics and safety. Sometimes those things overlap, sometimes they don't. And so there are other things to consider. I think the application domains have to be compelling, and I think there has to be a really strong argument for why we're going to do that. But yeah, I do see that as being a future, absolutely. I think that it's where these applications are compelling enough, there is really no reason why we shouldn't be, would be my view.
Fingers crossed. That's what Brexit happened for, right? Then you're just detached from the EU and all this regulation. What was it?
I'm not sure that was I'm not sure that was the reason Brexit happened, but we could get into that. There's still legislations, and there's still good reasons to want to be safe around this kind of stuff. The thing about biotechnology is I think it's one of the more ethically aware fields that I know. I mean, just genuinely does that. If you think about all of the kind of weapons building that we've done over years and these incredible technologies that have become rapidly dangerous, and you kind of wonder why the ethics committees were there. We've never put in a synthetic biology project together where we haven't had a work package looking at the ethics, public acceptance, safety measures, all of those sorts of things. They're baked into everything that we do now. So I have a lot of optimism that we're in the right place. We do need the legislation to catch up with that. Yeah.
Okay. Well, let's hope so. And with all the legislation in place, then are we going to go grow buildings from seeds? Is this, because you've also mentioned, I'm going to the next example of a project that is happening here with people like Dylan Oskan. So what's that going to look like? Because you quote this really nice book, actually, sci-fi book, in which I think Shenzhen or some Chinese city is described as growing into the air, literally, as we speak. So is this what you dream of at night? These kind of cities?
That's, I've forgotten the name of the story. The Pocket Full of Dharma is the name of the story. And I can't pronounce the name of the author. He's one of my favorite science fiction authors, but you may have a better...
I'll try to look it up.
Yeah, if I look it up. So actually, it's a Shenzhen is being grown, but also the growing buildings are also kind of metabolizing the old city as well. So in this story, they are cleaning up as they go this crazily polluted industrial city, and they're kind of moving through the landscape, eating it up. Yeah, I think about those sorts of things. I've even tried to write the odd science fiction short story unsuccessfully in this area.
Unsuccessfully? Why?
Well, because no one wants to publish it.
Oh.
I'm a better academic writer than I am a science fiction writer. But yeah, so there's that. It's interesting. I don't know. This goes back to this agential materials idea. I don't know that there is a way of necessarily imagining a completely self-assembling city in that way. What I do see is the possibility of self-assembling buildings that are under some kind of control. So the vision would be actually that you get... So for example, you can go back to kind of ancient examples of cultivation of plants. And this is something I'm interested in at the moment. I've been looking into. There's some really interesting, there's again rope bridges in India, for example. There are these, what you might describe as kind of almost extreme cultivation techniques like bonsai trees and so on, where you're kind of shaping the biological matter in particular ways. And I think this is the way that we might go. And so you have, for example, microbes that can create really interesting materials, but they need a lot of help, and they need a kind of scaffold or structure on which to do that. Getting into the mycelium work, which is the work that you were alluding to there. You've got with mycelium, this is a root network of fungus. You've got amazing material forming capabilities, but they are shape takers rather than shape givers. They don't have a form that is outside what you create for them as a scaffold or as a mold, or we're interested in, for example, chemical diffusions that give them different properties in different parts of their growth. So you're in this kind of dialogue all of the time with these external methods. I'd like to get beyond just molding them into blocks.
Yeah.
But I think there you're never going to get a mycelia that will grow into a building. But you might get really interesting, functionally different materials from mycelia by engaging with the way that they grow.
Yeah.
And so I guess the bigger vision of that is one in which construction is kind of informed from the outside. Possibly by some form of digital fabrication, by maybe robotics, which are giving the biology certain chemical signals and triggers at particular times. A synthetic biology layer, which will make the biology amenable to those inputs and outputs. And then that cellular capacity to build things without needing to be given molds and so on. So I think there's somewhere in that I see the future going. We may get to the point where we can plant a seed and have a building. But I think it more realistic, even in the long term, to consider a kind of symbiosis of human, digital, and biological construction working together as one.
Why not plants, though?
Well, I've just come back from a conference.
Okay, this is the inside information.
Yeah, so I have a couple of projects on the drawing board on plants potentially. The reason we didn't do plants is partly because I was learning synthetic biology, and you start with microbes. We also have really good microbiologists here, and so we started there, and it's a foundational starting point. I also understand the limits of what microbes are able to do without all of that external scaffolding and molding and all of those other things, and just in terms of scalability. Plants are super interesting. From a synthetic biology point of view, it seems like they're a nightmare from everyone I can speak to.
As if mycelium is easy.
Yeah.
Already saw plants.
There we go. So plants becomes a different thing. I think an agential approach to plants might start to become interesting though. And so I've been talking with a couple of collaborators about doing something around bamboo, largely because bamboo will just grow quick enough. And so that's the other thing. I'm kind of impatient. We need to get data, and a lot of the plants that we might want to work with grow very, very slowly. But bamboo is an interesting one, and would give us some interesting constructional materials as well. So yeah, plants is definitely an area that we're opening up, but it is, from my understanding of it, orders of magnitude more difficult to work with plants at the scale that we were interested in, and than it is to work with microbes, right? So that's why we're not there yet. 5 years' time, I think we'll have a couple of projects on plants, I'm sure.
On plants,
Yeah,
okay.
yeah.
So should we, but then do you see, for example, that we could grow something that you just plant a seed? I mean, the tree grows in a particular shape because it reaches towards the light. So
Yeah.
for example, if you had genetic control that is dictated by the physical conditions that you impose on these structures, then you're kind of co-creating a building, perhaps, that these tree will just grow in a box or in a nice shape.
Yeah, well, so there's a really good project called Flora Robotica that was led by Phil Ayres in the Royal Danish Academy in Copenhagen. So I think if you're asking about groups doing these kind of things, that his group would be very high on my list. And they were just using the natural ability for a growing shoot to follow the light. They've shown they've got images of plants that they've grown by just moving the light around, and they've actually got it to hit marks by moving the light around and then getting it to grow in those directions.
Wow.
This is an example of that agentiality, actually. So that's just using a natural input, if you like, that the plant has in its developmental pathway to grow it, right? And he will quote very old texts on growing and how people have done in ornamental gardens and so on, have grown architectural scale structures by cultivating plants in particular ways. My view is that there is a molecular layer there that we can look at as well, a cellular and molecular layer, which is to say that can we start to interrupt with or enhance the morphogens in particular patterns in a growing plant. So that's where I would like to get to. And I think there's some, without giving too much away, there are some relatively simple experiments that we can do with a plant like bamboo to demonstrate that for a functional end. So my interest would be to create a material that was what they call functionally graded. So it changes its material properties throughout its section to allow for a particular function. So if you think about a bamboo cane that could be more bendable in one place than another, where you've got different levels of rigidity. This relates to someone like Neri Oxman, for example, who's worked on this in 3D printing, where you create these functionally graded materials just by altering a mix within your 3D print through a surface or whatever. Biology gives you that kind of capability for free. It's just that we can't really access it yet. So my thinking is, how do we access that layer of developmental control such that we can make these functionally graded materials? So we've got ideas there. It will be tricky to get it funded because they're quite early stage. We don't have any preparatory data, but I'm convinced from the conversations I've had that there's a direction that we could take.
I mean, I'm sure all the plant biologists would die for this, really, because plant biology is really going through such a downhill in terms of investment, and IGEM, among others, is trying to get it back on board because there is very little money in plants and bio and in general applications of plant genetic engineering, because all of them are just used for food, because that is the thing that we just tend to think about when we think about plants. But realistically speaking, we can, really do crazy stuff with plants. It's just really hard. But
yeah, a lot of the research there is just in sort of optimization of yields and resistance to bugs in the environment and that kind of stuff, which is important. But I haven't met that many people thinking about plants as a fabrication platform. And actually, when you talk to the plant biologists, they get quite excited by the idea. I think there's ways of going there that are really interesting, but it will involve a bit of a mindset change. And I thought also finding the right funding is quite challenging for that kind of work.
Yeah. So now I'd like to talk a bit about bacterial cellulose. What are your thoughts on bacterial cellulose? Are we going to all be wearing things from bacterial cellulose leather?
Yeah. Yeah, I mean, so we see it as a sort of, I think we described it, I don't know whether we described it formally in a paper, as a proto-material, in the sense that, so we can talk about plants, right? So plants are amazing architects of cellulose. So one of the images I show in my slide deck is one of plant cellulose versus microbial cellulose, and this kind of amorphous mat of polymers that you get from bacteriocellulose versus this incredible structured cellulosic composite, essentially with hemicellulose and lignin in wood. We think that, though, it's an amazing kind of scaffold material. So you can either work to organize it better in its formation. So organize it, I say better, in ways that have functional benefits, or indeed embed other materials in it, through it, and outside it all. And also use it as the basis of an engineered living material as well, because it's a very good protection for microbes, for example, from the environment. We've got a few different projects in cellulose, the main one being one called Living Manufacture, which is just asking the question, how can we better fabricate it So bacterial cellulose generally goes as the result of a fermentation. It forms a skin on the top of a liquid culture of the bacteria, which we can now, using our bioreactors, grow to some thickness. So we've got about 8-9 centimeters thickness so far. We think we can get it much thicker as a continual kind of volume of cellulose. And we then modify it as it's grown. So we have a liquid handling robot which can drop modifiers into the cellulose as it's being grown. We can also add in other bacteria, which then also have modifying. So we work with an optogenetic bacteria, which means light sensitive. Yeah. So we can shine a light at it, and then it will produce a pigment in response to the light. So we can shine the light, different light patterns as the cellulose is grown to leave a trail of patterns behind.
Did you try to do the microbial carbonate precipitation as well?
We've not done. No, we've not done that yet, and that's definitely an area I'd like to do as well. So again, the idea being to create a functionally graded material, you can imagine that parts of that material might be calcified so that it would be really strong, or very compressive, have high compressive strength in one area, be very flexible in another.
Yeah, because then you can see the 3D structure of bacterial cellulose like a mold, right? Then you just shine light on it as it's growing, like in a vase shape, as you do on the renders usually, right? And it just precipitates, and then you can wash off the bacterial cellulose, and then you're just left with this solidified thing.
Yeah, I mean, to get to that level where you've got the calcium carbonate forming crystals that are going to be stable enough to do that with, I think is a huge challenge.
Open call for PhD students.
Yeah, maybe. That would be, it's more than a PhD project right there. But yeah, we know that the calcium carbonate, we've grown calcium carbonate in cellulose. And actually, interestingly, it does gets templated by the cellulose. So you can see the different shapes of crystal in there. But yeah, that would be the idea. And so this is the idea, again, of bringing a kind of robotic fabrication together with the self-assembly of the material. So it's like, we call it like a 3D printer. But actually what you're doing is you're growing a functional material in that way. I think there's a lot of potential there, and there's loads of ways you can make it more complex. And so that's what we're trying to get to with that.
Okay. And I want to step into the philosophy behind this, really, because all these are really exciting examples, and there's many, many more projects happening at HBBE that are super exciting. But when we zoom out and consider the broader vision, how do we make sure that people in the industry and people who walk on the streets and look at the buildings that we're trying to build here still think, oh, this is a building, this is something that I can step into? And these are some projects that I know our colleagues here at HBB have been working on, with projects like the living room, for example. It's a project that Jane Scott has worked on. They create a knit that they then depose a layer of mycelium on, then the mycelium grows over the knit, it solidifies, and you have a solid structure. And they made it into like a habitat. So they're exploring, how do people feel in such a space? And the spaces feel very, very different. And perhaps this is actually really interesting for me to think about personally, because it creates this, how when you have these regional architectures, they all feel very different inside. Like a castle, even here in the northeast of England, looks and feels very different than a castle in the south of Spain, or an old house in China, for example, looks very different than one in rural France. So is this perhaps something that we can imagine, that we're going to bring back these vernacular architectures grown with local mushrooms, with local materials that have different acoustic properties, different tactile properties, perhaps, that we're going to be occupying in the future? And this is all going to bring back the diversity that we're losing so rapidly with our globalization. Is that something that you align yourself with?
Yeah. And the living room is an interesting example of this, right? So, I mean, aside from the charismatic nature of that installation, and it was very inspiring, particularly for children, actually, that they seemed to be really taken with it. And it had a real reputation in that exhibition. She also used wool, which was waste wool that had been sourced from the Lake District. So just, I don't know how many miles, but not far, like an hour's drive away from here, an hour and a half. And the mycelium was grown, I think I'm right in saying, on waste paper as well. So waste paper is the major source for the mycelial growth. So it was really interesting from that perspective, as well as scaling up. And actually, Jane is one of the few people in the HBBE who's really produced things of scale so far, and a lot of them at this point. A lot of our materials are still frustratingly small. And it's very difficult, as you point out, to engage with things that are barely on a lab bench, sometimes down a microscope. And so this is a mission that I've set the group that we have to scale up. So this is one of the things that my team are thinking about at the moment. What is the most scalable materials that we've got and how we can build something interesting with them? There's a great tradition in architecture of building pavilions, and I think this is the way to start. The broader context that you described, well, we called it, there was a paper that we wrote probably maybe 10 years ago now called Radical Vernacular, which was exactly that point. But this was about space exploration, actually. So this is very much influenced because we do work a bit with NASA, and you mentioned at the beginning that the space exploration is this. There's a kind of one where like NASA will dream those dreams because you can't take cement trucks to space. So how do you make materials? How do you get them to self-assemble? All of those sorts of things are very present in their thinking. And so we talked about a Martian concept that we had using biominerals. It wasn't super original, if I'm honest, because I think there's other people who've done it as well. But we described it as a radical vernacular. You just literally use what you can find around you, and that's it. We've gotten approached by lots of people who are interested, particularly the construction sector is very interested in what they do with spoil from construction. Because it's crazy that you move a volume of earth away and then you bring a volume of sand from across the world to your site. It makes no sense, and they know it makes no sense. So finding ways of dealing with those very local resources is important. And what biology is really great at is turning all of these kind of very simple molecules into very complex molecules and structures. The tree example again, the roots grow into the soil and get everything it needs to grow, the cellulose and so on, other than the energy from the sun. You've got those two things, and you've got then the basis of a very sophisticated construction. So that would be the absolute ideal. And as I say, we called it radical vernacular, which is essentially you draw a line around your site. So I can only use what I can find here. That would be the ultimate, right? So that would be the way to go. There are probably some steps in between getting from here to there. I mean, if we just describe biology as a technology, and there's a philosophical problem with doing that, but I'm going to do that anyway. If biology is the only technology that could do that, I don't know of any other technology that can turn Earth into trees. And then so there has to be a biotechnology solution to that problem. I don't see another way of doing that kind of monumental magic, really. So that for me is the basis. This radical vernacular idea is the basis for a lot of what we're trying to do in biotech.
Yeah. And I have a speculative question about this. When we're considering growing buildings and instead of constructing them, what do you think the regulation should be? Because I've been having these impulsive thoughts while being here at HBBE that we should just go into the field and hijack any structure that we can and just grow stuff on it to spread the word about it, you know? Any like carcass. I just passed by in Oosburn. There's a really nice carcass of metal tubes standing there, completely abandoned. And I was looking at it, I was like, could use some mycelium or bacterial cellulose things on it. It would be cool, right? And then people would be passing by. We can put a board there, just being like, this is the stuff. This is what you're going to be living in. And that's it.
Yeah. I mean, so... Well, the kind of guerrilla living architecture.
Yeah. Because it's not regulated, right? So no one... I had a conversation with Ben, actually, about this, where he said, I have in my backyard a 15-meter tall tree.
Yeah.
And no one can tell me to take it down. If I were to put down a structure from concrete, for example, or from whatever, from mycelium... No, well, from mycelium blocks, I actually don't know, but from bricks, let's say. Yeah, my neighbors would come. They call the police, and then the police would come and say, you don't have a permit to set up here. But for a tree, you don't need a permit. Meaning that by transitivity for mycelium-based materials or for plant-based materials that you grow into particular shapes, you don't have a permit right now.
Yeah. I mean, I wonder whether that's completely true in the sense that if your roots are encroaching on someone's garden and you're undermining their house or what, I wonder whether there are ways in which there is a kind of resource.
Yeah, maybe, but under the train line in Oosburn, under a bridge,
no one cares.
Probably not.
Yeah. No, that's interesting. I hadn't thought about doing that. That would be
Bacterial Cellulose Summer School 2024. This is it.
I suppose then there must be, legislatively, I don't understand what the difference would be between a human construction and a natural construction as well. And that's the line, right? So I think bringing our own biological materials in this way onto site, that constitutes a construction in a traditional sense, even if it's a non-traditional material.
I'm not sure. We should look into it.
Okay, you can see whether there's a loophole so you can exploit here. I think I mean, there is a pragmatic issue at the moment. I spoke to someone at Ecovative. They make mycelial composites. Mainly they do it for packaging, I think. But they do have, I think I'm right in saying they have some patents on an insulation material product. And I said, well, you've not really done anything with that. And they said, no, we just looked at the legislation around construction and we just couldn't we didn't want to go down that route. And so that is a barrier at the moment. I mean, I feel like when it comes to construction technology particularly, we also need to rethink our ideas of tolerances as well. And so much of our industry is based upon standardization. It's very difficult to get standardization from biology. And so that question over what standards we have and how we achieve standardization, I think that's going to be one of the big issues. And so we're talking Monica, who has been doing a lot of the space architecture stuff, and she's been making these components, and her failure rate is massive. And our collaborators have kind of come back and said, well, if 1 in 5 of your components is going to fail, this is no use to us. My view of this is you just have to have a construction technique that allows you to have 1 in 5 of your components fail. And that's possible. With concrete construction, there are failure rates. My grandfather used to do precast concrete stuff, and you can only, no 2 beam would be the same. You just have to make sure the tolerances are such that it's not going to collapse, right? But I think we need to rethink that in biology, and that has then in biological created materials, that's have a knock-on effect into construction and how we evaluate buildings. And so I think that's the area I see more challenge.
Yeah, but I feel like, but this is why I'm bringing up this guerrilla techniques, because I feel like somebody just drew a line, said, okay, if it's not up to the standards of steel and concrete, you are not coming in. And indeed, that means that for us, we are already restricted on so many levels with biotechnology. We can't do any GMO on plants in Europe, et cetera, et cetera. So when we're trying to reimagine things for radical sustainability, somebody says it doesn't compare to these horribly detrimental materials. Therefore, we're not going to even try them. And this is why I'm saying that, okay, well, let's just start doing it, and then somebody's going to take a look and they're going to be like, well, this is a house standing, and there's going to be more public acceptance. Because right now it is indeed only on the level of, okay, let's open the material ecology book by Neri Oxman and take a look at this beautiful Hoya structure, and et cetera, or look at Jane Scott's work in a gallery. And it's still not, it's not outside. And I think this is also an interesting aspect to talk about maybe briefly, because I know you think about this, is dirty engineered living materials. Like, how do we... Because right now we're talking about biology and architecture and microbiology and architecture, but everyone knows who's ever worked with bacteria or brewed beer or kombucha or kimchi, you get contamination really easy. So how do you grow these materials in the wild? This is a really important example. Also, since probably if you want to use GMOs in the wild, you want them to die very quickly, and that means that any other species will outcompete them.
Yeah. You don't want them to die very quickly.
Yeah, you don't want them to die very quickly. But if you put the kill switches or something like that, right, then they have a tendency to destroy themselves or be less competitive, to not survive in nature, to bioethically protect us from those GMOs in the wild.
Yeah. An interesting concept that I took from the agential materials stuff was that, so in developmental biology, the flip for me in thinking about mammalian cells, for example, as agential, is to think of a multicellular organism as essentially as a swarm of cells which are collaborating, sometimes competing, but often collaborating in order to form structure, people and mammals and everything else, people and everything else that they do. The other side of that is, if you think about microbes as single-celled organisms, we tend to do what we tend to do in synthetic biology, which is to take them out as, our E. coli or whatever in a dish, and we work with them in that way. But actually, you can, from an agential perspective, they're just distributed multicellular organisms, right? So what you do, you see in, say, a Winogradsky column, which is one of these columns where you grow these microbial communities, and they essentially self-organize into different colonies doing different things within that structure. And they can be self-sustaining for long periods of time. We have a couple in the Ohm, our experimental house, that I think have been there now for 2 years and ticking over, doing their own thing. What they've done is essentially to assemble vital organs for that system. So there are different communities doing a different job. If you took out a cell from, as developmental biologists might, you took out a cell line from an individual and just try to grow, I don't know, whether it be liver cells or kidney cells or whatever, just on a plate, there's a limit to what you can do, right? Without the companionship of all of the other organizing cells they need around them, they can only do so much. Similarly with microbes, if we take them out of that context, there's only so much they can do. So I think there is definitely a role here. And when we call about dirty ELMs, what I'm meaning is really engineering microbial communities.
Yeah.
And often you might be only doing the engineering bit on one part of that community, but recognizing that it requires those resources and that context in which to operate. Going back to complex problems, it's a really complicated problem. It doesn't fit well into a schema of simplification and modularization of parts and the construction into more complex wholes. You've got a complex whole to start with, and you have to try and reverse engineer it or unpack it in some way. That, for me, is the only route towards engineered living materials.
Yeah. Does this mean for you that there should be a paradigm change in regards to how we think about biological systems and engineering them? Because indeed, as you poignantly stated, in SynBio, we have this approach that is very similar to transistor. I mean, it is exactly taken from that, in fact. But in biology, we take things out of the system, you put them back, they don't work, right? The hierarchies of emergent complexity and in general emerging systems don't work the same in biology because of the agency, right? Because these are living things that think for themselves. And I'm not even sure the problem could be alleviated if we overcome the challenge of minimal cells. Let's say if we are completely synthesizing de novo the cells by putting some minimal components. I'm not sure if it's going to be manageable to work with it as we do with computers.
Yes, I completely agree. And I think there does need to be a paradigm. I wouldn't want to be super critical of synthetic biology as a field, because actually it was my gateway into this. And one of the things that synthetic biology has done actually is to make certain biological concepts a lot clearer and a lot easier to understand for people from the outside. And so teaching it in this way is incredibly useful. I'm not sure I would have been able to enter the frankly Wild West world of molecular biology without synthetic biology as a starting point, because it was a set of principles from engineering and design I can understand and apply. And it's been incredibly, it's created an amazing community, and there's been some real breakthroughs there that I don't want to discredit those. But I think you're absolutely right. It's only one level of the biological system, is the genetic level, right? And so there are many other complex interactions. There are many other aspects of biological function that give, for example, form to a tree. So one of the reasons I don't think you can grow a tree into a house is that whenever you ever seen two trees that are the same, they are plastic in their nature. They're constantly responding, adapting to highly complex situations, both internally and externally.
Yeah.
So we need to understand those additional levels a lot more. And we're not putting any time into that. So the plant biology thing is an example of this. Yeah. I don't think that genes are the right interface for plant cells to be able to do what I want to do in fabrication terms. I just don't think we can do that.
Okay. But do you believe that these kind of technologies will help us to decentralize the fabrication, or are you really trying to develop technologies that are plug and play for the large industries? Because we could think about building sheds and kind of like on the scale of just growing a shed with grown mycelium by doing like a DIY kit, which can easily be self-assembled in your backyard, let's say, and then that's how you do it instead of timber or instead of other materials that you would consider.
Yeah. I probably think a little bit too conservatively about this. I see certainly the first generation, second, third generation, I don't know what generation they would be, but the generation of fabrication technologies that I see coming online as existing in something rather more like a factory setting to start with. There's something a little bit more like the kind of environments that we already work with. It's interesting if you look at I guess this would genuinely be the first generation of biomaterials. So think of Ecovative, for example, with their mycelial packaging and the mycelial foams that they're working on. Those are factories, right? They're factories that happen to be growing things, but they look and feel like quite traditional manufacturing contexts.
Yeah.
They're pushing it one step beyond that. But then I think there's another step beyond that, which is to say that I can imagine factories that are more like robotics plants that make cars, for example, now, but rather than making cars, what they're doing is influencing biological systems, these programmable fabrication systems. I do see that being quite centralized. I see that being part of the existing kind of environment. The ultimate expression may be this kind of radical vernacular view, which is essentially then we have these biological assembly systems that can be used by individuals in their own gardens or their own plots of land or whatever. I think that's an interesting possibility. I don't see the route to that quite as clearly. I mean, Biodesign has a DIY bio community working in this area, of course. So there is that kind of what is it? The kind of the car garage kind of do-it-yourself kitchen kind of mentality going on. In synthetic biology, there's been a bit of that with the DIY bio community and the sharing of lab resources and equipment and those sorts of things. I think we're a long way from that. For me, I still think that I see the route towards these kind of materials being at least starting with something that looks a little bit more like the world as it looks today, and incremental changes in that direction.
Okay, so you think basically first it's going to be factories using these technologies, and then so it's not vice versa.
Yeah, I mean, if you look at the investment in biomaterials at the moment, so for example, there have been investments in different biopolymers and so on. The ones that seem to be getting the most funding are those that are able to adapt their materials and their processes best to the existing industrial infrastructure.
Well, of course, yeah. I mean that makes a lot of sense, but for me, it's just I'm not talking about. What I'm asking you is really about the vision, not about doing the most economically possible and feasible. Of course, if you do plug and play solutions, if you make bricks from mycelium, it's easier than to tell some construction companies, oh, you know what, I'm gonna come here with my wool and I'm gonna put it down and I'm gonna give me 2 weeks and it's gonna be a solid structure magically, and people are gonna love it and it's gonna be cool, insulating, fire retardant, whatever. Like somebody is gonna look at you, gonna be like, you're crazy, what are you doing? But if you say, oh, I have a mycelial brick that you can put down, they're gonna be like, cool, let's do it.
Well, I suppose that I'm always in the middle here, whereas I think there's a difference between the vision and the strategy. So I think that, there are some amazing thinkers who have that kind of vision as well of a built environment future. For me, I don't know what experiments we can perform that will, if I set that as my goal, it will get us there. So my first thinking is I need relatively controlled environments to do a lot of the growth and the more sophisticated stuff that I want to do. Yeah. So it makes sense that in the vision, I'm mapping that onto an enclosed environment where I have some level of control to start making things. And so the vision to kind of drives it, because even that is a fairly, far out kind of concept, but at least we can start to create within our own labs and so on, the environment that might become, and sell that to people who kind of understand that environment already. There may be a more radical approach to this. The problem is that I don't know what the experimental route is to deliver it. Right?
Yeah.
So that's where I always get a bit challenged with that, as I say, you can be the visionary in this area, but do you have the strategy to make that happen?
But I mean, it only takes a person, really. When Steve Jobs came around, he said, I want everyone to have a computer. I want everyone to have a computer, and I want it to be as accessible as possible, right? So user-friendliness has been at the core of Apple's ideology and philosophy behind designing their technologies, right? And back in the day, and their whole marketing was about that, like in the 90s against Microsoft and stuff, that this was the way. But maybe in synthetic biology, what we're talking about is, let's put it in a factory, let's make it for nerds. But in fact, maybe we should make it as accessible as possible and make it plug and play for people to put in their backyard and be growing, kind of similar to how everyone is growing kombucha and kimchi and stuff like that. You say, oh, okay, well, let's do like open source DIY 3D printers, or similar to your living manufacturing system, that you could be designing all sorts of objects and shapes that have an inherently different aesthetic and feeling to them than doing like PLA based 3D printing thingies, because there's such a big community of people who would enjoy that, I feel like, and especially since it has that living aspect, it would fascinate so many people.
living manufacture is interesting and is different in this regard, in the sense that we've used quite a lot of open source hardware, and we're going open source with the build, and I do have that vision that everyone might have a living manufacture device on that they've built on their own on their desks, and it's been deliberately designed to be accessible in that way, and it uses pretty much off the shelf components, albeit configured in interesting ways.
That's great.
I just want to pick you up on the Steve Jobs example there, because this is a question that I've had. So I intimated before that one of the reasons I got into the biotech thing is because I thought, like, well, computing is, I mean, I should have gone to AI at that time, of course, but computing is kind of like, it was almost seemed a bit old school. This was being sorted now. This is an emerging technology. It was kind of already in, whereas biology wasn't. The question for me is because I've been reading about the kind of early days of computing a lot at the moment, is about whether biotechnology is at the Steve Jobs point, because Steve Jobs and his kin were riding a wave that was already happening. So I think arguably, if it weren't Jobs and Gates, it would have been other people. It was the right moment, the right set of ideas, but there were other people who were very close behind in terms of those ideas and similar thoughts.
Yeah.
Whether in biotechnology we're at the point at personal computing was, or whether actually we're still at Charles Babbage.
Yeah.
Right?
Yeah.
And there's some days when I'm speaking to people who've been in the lab, I feel we're at Babbage, right? We still don't have a calculating engine yet.
Okay.
So and then the vision becomes a fundamental one, because the Babbage vision is calculating machines and so on. It's a different path, right? So I don't know quite where we are with biotechnology now. I'm kind of interested because I joined thinking we're at the Jobs point, the sort of Jobs Gates of personal computing. I don't think we are at that point with biotechnology yet. I don't think we're there at all. I think we're still quite conceptual.
Well, I hope it will develop rapidly. We have all the technology.
Well, we do. I mean, it's helped by computers. I don't want to, computers are kind of useful in this regard, right?
Yeah. All right. I think to wrap up, we have our traditional question for you, Martyn. It wasn't we didn't want you to prepare for it.
Right.
But seeing the state of the world in terms of sustainability, in terms of rigidity of particular regulatory institutions, legislation, and the industries themselves, and you being this cool kid who is engineering stuff with very, very pioneering, trailblazing technologies and thinking ahead.
I'm mid-40s, by the way.
That's okay. No, they can't know this.
I have a bad back, as I explained to you before.
You're spoiling this to the listeners.
Sorry.
Know what you look like.
Okay.
But what gives you hope?
Well, I mean, going into we have our research group meeting tomorrow. That gives me hope. I mean, generally, it's exciting. I've just advertised a PhD studentship. I had 18 applicants, and 16 of them were eminently hireable, incredibly passionate, had often done this whole biodesign thing in contexts where there was no biodesign. We were writing stories about how they'd gone and found a lab and done these kind of things. There's so many people like that now. And since the HBBE, we've had so many more. There's just this groundswell. And I will go into my son's school, as I did. My son is 8, and I went to deliver to his class. And I asked at the end whether they were asking questions all the way through, frankly. But I asked at the end if there was any questions, and one of the kids just said to me, I'd heard that mycelium communicate through a network underneath the woods, and they create this. He knew all of this stuff. That I've been trying to persuade my undergraduate and master's students to learn. That's hope. That's hope, I think.